Okay. Okay, everyone will
So,
go ahead.
can everyone see my screen?
Yep.
Okay,
Yep.
great. So, hello everyone. My name is John Polanco. I am originally from the Dominican Republic, and I'm currently a senior TA here with PPCR. I've been with the course since 2018 when I was a student, and then I've stayed with this amazing team of people as a teaching assistant. I'm currently completing a Master's of Public Health at the Harvard T. School of Public Health. And today we'll talk a bit about Introduction to Clinical Trials. I have no conflict of interest to disclose in relation to this topic. And so before we start, I just want to say this is going to be a very interactive session, and I expect everyone to participate as much as possible. If you have any questions, what we'll do is we'll divide as we'll see next, we'll divide the lecture in two sections. One section for a case study and then practical applications. So if you have any questions, please write them down, and then we can talk about these at the end of each of the sections, but we will interact throughout the lecture. So this is our agenda today. We are going to have a case discussion from 7 a.m. to 7:45, and then from 7:45 to 8:30, we have practical cases. So first things, in case that we run over, I know that many of you have a very busy schedule. We don't expect to run over, but in case that it happens, please feel free to drop out of the class. These will be recorded and you can easily access it. And the objective of today's lecture is basically to promote critical thinking and an in-depth appraisal of the case study that you were assigned for this week, and also other practical cases that we will develop as we move on the way. Okay, so let's first start with this very basic question. And what is research and why do we conduct it? And I don't know if Keiko can help me selecting one of the students.
Sure. Maybe this is the first interaction for us. So I will choose someone from Japan. So Kento-san, can you open your mic and answer to this question, please?
Yes, my name is Kento Ikegawa from Japan. The research is conduct new things and new drug.
Yeah, why do you think that we should conduct research?
It is important to understand ethical things and past and research.
Okay. Yeah, and you bring a very important point here, is that we use research to develop new things, and it's we can formally define it as a process where we want to respond to find an answer to worthwhile scientific questions. In that scenario, it could be a drug, as you mentioned, but it could be other interventions, not necessarily a drug. We could find the missing bridge in certain biological, let's say, biological processes, and we want to find an answer to that. And so very important for us to remember that when we conduct research, we do it in a systematic way so that it is reproducible. And we use a scientific approach as well. And then research literally means search for scientific knowledge when we are applying it to biomedicine, etc. And systematically is a formal process to increase the fund of knowledge that we have and we use properly for the development of novel applications, be it that we want to find a new intervention, a new drug, etc. And then we have different types of research. Why don't we get another participant to chime in on the different type of research that they know and their function.
Maybe continue. Kento-san, can you continue?
For example, a historical control study and RCT, prospective study, that's all.
Okay, so can you, in a bigger category, can you group all of these research like designs that you've told me? Where will you use these when you're doing these studies? Let's just ask another question. Do you think these are only for human? Are only applying humans? These type of designs that you told me, or do you apply them in other kind of participants, or non-human subject research?
You mean animal study?
Exactly. Okay, animal studies, and if you had to before the animal studies, the one that you previously mentioned, if you had to group them all together, what would you call them?
I don't know.
Okay, it's okay. So can we call upon someone else?
Clinical trial?
Yeah, could be a clinical trial, as you mentioned. That's one type of research design, but these are normally used in, okay, let me see, we have another participant, Shara Faldil. Faldin, I'm sorry if I mispronounce your name, please.
Hello. So I believe we can look at the research from multiple perspectives. We can look at it through quantitative, qualitative. We can look at it from a prospective or retrospective. We can look at it in animal studies or human studies.
Perfect.
And through the different stages of the researches as well, like phases, phase 1, phase 2.
Okay, perfect. Okay, and as we were discussing here, those that were previously mentioned, we could group together in what we call clinical and population-based research. And the animal studies are usually pre-human research, and these are not considered clinical. And so we have three basic types of research, and these are basic science research, translational research, and clinical and population-based research. Can anyone tell me what they know about basic science research? Any ideas that may come up when you hear these words?
Anyone from chat? Anyone? Volunteer? Please open your mic.
Good morning, everyone.
Morning.
I believe this basic science research is when we go to the lab and we are going to look for a tissue, for example, and you're going to look for the basic of it. In the lab, how it works, how it's functional, really the basic, how, I don't know, a tissue works or a movement works or the basic. And the translational research is when you're going to try to apply this basic knowledge that you got in the lab, in the bench, and you're going to try to put in practice. You take it, for example, for a new drug, and you're going to put that for the patients. So it's from the basic in the lab, and you're going to translate it to new treatments, new drugs on the practice. And you can do that by using clinical and population-based research.
Excellent. Great response. So to summarize what we just talked about, basic science research is not applied research. You simply go and try to understand, let's say, normal versus disease. What's the mechanism? How things works. So now that we have the basic foundation, we want to say, okay, we have that, but is it applicable to humans? Do we want to? How do we translate from the bench to the bedside? how does this apply to humans? So that's what translational research mostly concerns itself about. And then we have clinical and population-based research when we really try these in humans, okay? And then another question is, what is a clinical trial? Anyone else?
Can I
Hello.
call?
Good morning.
Please, go ahead.
Okay.
Ibrahim, can you name first? Could you please say your name?
Okay.
That's the first. Okay.
My name is Hernan Agoro. Okay. A clinical trial is after we made after we have our project or clinical research, all our basics. Now we're trying to apply until trying to figure out all the phases into phase preclinical to the phase 4, following all the project outcomes and aims.
Hi everyone. Can I answer? It's a set of procedures in medical research that try to develop a secure way to do research in human subjects, not the treatment of the patient.
Okay, perfect. And then let me just ask you one question, Daniel. Now that you mentioned, what do you mean by not the treatment of the patients?
Because the main focus of the clinical trial is not treat the subjects. It's just acquire new knowledge about the treatment, this new drug, for example.
And that's very important. We'll talk a bit more about that later on in the lecture. And when we talk therapeutic misconception, we will introduce that concept. But thank you very much. And you have mentioned a very important point here, and is the fact that human trials, clinical trials, are conducted in human subjects. In these trials, we evaluate a particular intervention, and as you've mentioned, to see whether it is applicable, it works. Depending on the phase of the trial, there are, of course, many different phases. And lastly, another participant, if you can please share what motivates the conduction of a critical trial.
Cecil, can you go ahead?
I can answer. It's Adriana.
Anyone? Okay.
So I think that what it motivates clinical trial is at the end to gain knowledge about something that we want to do. For example, we want to try a drug, a medical device, or something in humans. So you need the clinical trials at the end to see efficacy, safety, and effectiveness of what you're going to try and new.
Yep, perfect. Thank you for sharing these, Adriana. And we can actually summarize your ideas in that we want to, in a structured manner, assess clinical outcomes, or not necessarily clinical outcomes. It may vary depending on the phase of the trial that we are in, and we'll talk a bit more in detail. But in general, we try to make causal inferences, and we want to assert whether the intervention works in humans. And then, without further ado, we will go ahead and start with the case discussion.
John,
And let's first, yes.
John, sorry, Keiko talking. I found one question from chat. Do we have time? Sure. Okay, so Bruna, can you open your mic, or you cannot talk, Bruna?
Hi, hello everyone. I don't think I get the whole idea of translational research. I am relating it with the phase 1 study.
Okay, so phase 1 study is one part of clinical trials, and clinical trials are not considered translational research. Clinical trials are outcomes research, mostly part of population or based research. And sorry, let me just go back. So we have clinical and population-based research, and this is where clinical trials fit. And then we have translational research, which is basically you should not separate it completely from basic science research, because you're doing lab work. You're testing things in the lab, and you may do basic science research, for instance, to elucidate the mechanism of how iron is metabolized. That could be basic science research. But then you do translational research, and you say, okay, so how is iron metabolized in a human cell? How does iron gets absorbed in the intestine, for instance? These are things that get considered in a translational type of research. But then come clinical and population-based research, where you say, okay, now that we have iron, let's see how safe iron is in a patient, and let's try different doses of iron to see what is the optimal dose before I get toxicity or something like that. You know, you're applying this knowledge to human subjects in a sort of more applicable way. Did I get the idea through, or is it? Do you still have some questions about it?
No, it's clear now. Thank you.
Okay, you're very welcome. Okay, so I want 3 different volunteers here, and I want you guys to one of you to summarize the case study, and then 2 of you to give me your takeaway message from the case study overall before we discuss.
Okay. Okay. So anyone? So Fleming, you are raising your hand. Yeah, go ahead, please.
Sir, which case are we talking about?
This week's case study.
Yes.
Oh, okay. So that is the assignment that we got, right? In the lab.
Yep.
Okay. So we were at it was a hypothetical situation wherein we are at a lab at the University of Pennsylvania, and we have a group of scientists headed by Professor Geeks, and they're talking about a new therapy that they discovered that is in which they can probably use a lentivirus, and they can introduce a working gene into the lentivirus, and then they can introduce it into a patient who has a faulty gene, and replace the faulty gene with this new gene. And they have already conducted a few preclinical studies in animals, and they have seen that it works to some extent. And now they have also done a phase 1 trial, and they were discussing that whether this can be carried forward. Now there were some ethical aspects that were discussed inside the case study, wherein we see a very emotional doctor who is very emotionally and personally motivated, because he had his family members who suffered from some kind of genetic disorders. So he's specifically motivated that any kind of outcome that that may be yielded from the study would be useful, and he was ready to sideline the risks that the volunteers may face while being a part of the study. And then we have a new jet lag professor who joined. I forgot her name, and she reminds the whole team that sometimes there is a concept of therapeutic misconception that can be introduced into such studies, wherein probably it may be a lack of well-informed consents, as in if you enroll a critically ill patient into a study, they may get enrolled into the study just because they may think that they would receive a potential treatment at the end of the study, which is the wrong motive of being included into the study. So yeah, I think you can add on to it.
Yeah, great. I think you've made a great summary of this. Basically, the idea is that they're studying the hypothetical scenario, how to find a cure for HIV, and then they are faced with different approaches, and we don't say necessarily neither of them is wrong, but different ways to which they can approach this problematic, whether it is ethical to move on. And as we will see, there's also some problems that had already happened at the University of Pennsylvania with a very famous case called the Gassinger case, and we will discuss that in depth moving forward. And then two other students, do you want to share what your takeaway message or the main ideas are for this case study?
Anyone?
Go ahead, Graciela.
Hi, good morning, everyone.
Good morning.
Good morning.
Well, I read the text and they talk about the phase 1 and they tested on 5 people, of which 4 people reacted well and their T cells were maintained and increased. That means I think that the investigation works. And they don't mention anything about severe side effects, assuming they did a preclinical research, because in the case they didn't mention anything. The next things will be continue with trial 2, and that is where the discussion begins. Should we continue or should we stop? In my opinion, easy risk benefit evaluate and contemplate the 3 core principles: autonomy, beneficence, and justice. In this case, they will be respect. Since we obtaining the study participants, their consent will be asked and they will be told everything they should know about the study. It will be for your benefit. As explaining in trial 1, there were no greater risk with the 5 patients who were part in trial 1. Otherwise, the case explained by Dr. Helsinger, which despite the facts that were more risk than benefits, he didn't care and continued with the study. But in this case, that is not the case. For me, they should continue the study planning based on potential benefits, and if he sees any side effect that has not contemplated, stop it in that moment, do a retrospective study to find the error.
Very good, Graciela. You've made quite some very interesting points that we will definitely touch upon later on today on the lecture. And you have very well substantiated your choices here. And then I've seen that Pablo also had his hand up. So Pablo, why didn't you tell us what your takeaway message is?
Hi, everyone. So first of all, I found Professor Geek's experiment very interesting and all the opinions of his colleagues about it. And yes, it was like, let's say that all these researchers are finding these like preliminary results with 5 patients, and that even if the viral load and the T cells are getting viral loads getting decreased and T cells are getting better, Professor Geeks even states that these are preliminary results. And so I think in research we have to be really careful on what they find, because as I remember, when you use such a little amount of patients, in this case sample size, yeah, I believe we have to increase a lot the sample size, because when you have so little patients, you still need to increase the sample size, because you do not know if your results are just simply by chance or are really results that you're looking for. And so I was thinking about also that when they mentioned that they wanted to use, they first started using 1 infusion, and then I believe it was not adequate that they suggested that the next step we use 6 infusions. So my idea about that is like, okay, we can have proposed, like, instead of using 6 all at once, maybe you can try with 2 or maybe 3 and then analyze what will happen, especially regarding adverse effects with patients, and see if again this has the same efficacy as they showed in phase 1. So I believe you can be a little bit more cautious of what you can do on your next steps
Yeah, thank you for sharing your thoughts as well. Definitely, we could say that there is a small amount of subjects, and you touch upon a very important topic that is you may not have enough sample size, but as you know, and we will discuss that when we're conducting clinical trials, basically we have varying number of subjects that we may include in the different phases that is acceptable and is as a norm right now acceptable. I'm not saying that it is ideal to have this small number of subjects. However, it usually, as we progress in the phases of clinical trials, we do increase in the number of subjects that we include. And I see many people with their hands raised. I'll give you the opportunity to participate. We definitely have a more prompts to talk now. Here, and I want you now to tell me what you feel the characters. Can you define them, what their position is? Let's say Dr. John Geeks and their relationship. How do you feel their relationship is emotionally speaking with their research? Do they set their emotions aside? Are they very involved in the research emotionally speaking? So anyone want to take a and try to talk about Dr. Geeks?
Anyone volunteer? Maybe from...
Can everybody
sure.
listen to me?
Who's talking now?
Sorry.
Okay, go ahead.
Hi. Can everybody hear me?
Yes, yes.
Yes, Neeraj.
Go ahead,
I'm
please.
The question, what I wanted to say is, I think Dr. Stevenson is biased toward carrying on with the study. I would like to know if, I think, in my opinion, that it's therapeutic misconception working the other way around on the physician, because he believes that his research is going to treat patients. That's why he says no pain, no gain. I mean, so he wants to carry on with the study thinking that it will really treat patients instead of gaining knowledge on the research study. So I think he should not be involved as the principal investigator and trying to, he should be involved in recruiting patients, because in that he's obviously biased.
Okay, very good. And let me just ask you another question right there. Do you think that a researcher can do good research without passion, or what will you say about it? Do you think is it mandatory to have an emotional link about your research?
Yes, I think the whole point of doing the research is being, in some way, believing that it works and being passionate about it. I think that is the motivation for doing the research. However, it's hard for me to understand whether this person who is motivated to do the research should be involved in recruiting patients and, you know, that kind of stuff, because he's obviously biased, right?
Yeah, I mean, you can take different positions here. There's no right or wrong answer, but this is just food for thought. Where we like, if you're passionate about it, great. But to what point can you be passionate about it? You know, to what point may interfere with your ability to be critical about what you're doing? So thank you a lot for your comments. And I'm seeing that we have very great comments in the chat as well, where they have been describing what Dr. Geeks is, and also I see that here. I don't know, Penelope, do you want to talk about Dr. Geeks, Cameron and Wong?
Yes. Can you hear me? Yes.
Yeah. Yep.
Okay. I think Dr. Geeks, and I agree with most of my colleagues' comments. He's a dreamer. He wants to find a HIV infection cure, and I think that's amazing. The way he behaves, trying to remember the history, trying to avoid those mistakes as well, and try to make his team to critically think about the ethical implications of this study. So I think that's important as well. What I think is he has to find a balance between being passionate and being objective with the study. So he doesn't unfold in that slippery slope of the cognitive biases that sometimes we can fall into if we let our be emotionally bounded to those decisions, and then we can be very supporting of the technology that we are studying and not very aware of the possible limitations, and those could be harmful for the subjects.
Thank you, Penelope. And then, so here we can see the different players again, and everyone has done a great job. I see someone else with your hand raised. Do you want to?
Yeah. So can everybody hear me?
Sorry, I can't see your name, but yeah. Oh, yeah. Go ahead.
Yeah. So just one thing about Prof. Geeks. I think he had like a kind of passive aggression, because every time he was mentioning about his trial, he was reflecting like what could happen in his trial to the previous events that happened. Like he was saying, always remember Mr. X, what happens to Mr. X case, like the previous case that happens already. So I felt that he was avoiding mentioning that this can happen to his trial. And also when Dr. Steven mentioned about his first trial, that he was mentioning that it's very promising and it's helped decreasing the viral load and the increasing the lymphocytes. He was, I felt he was avoiding mentioning the possible downsides or the possible side effects that happens. As well as he was overdriven by his passion, as my colleague mentioned. I think they were trying, I don't want to say hiding, but they were not so honest, I would say, about what can happen in the phase 1 trial, like the possible side effects, until Dr. Cameron mentioned about this by the end of the case discussion. So yeah, I think this is what I feel when I was reading the case, that these people, they're trying to not mention what the downside or what the side effects of their gene therapy that they are studying.
Totally. Totally. Thank you for sharing your thoughts. And then one important thing here to know is that this is how it happens in real life, and this is why it is good to have different personalities in your lab to balance the different emotional like things that might happen during at a research meeting, and someone may want to move forward, some might not, but then you come to an agreement. And then generally speaking, we can define Dr. Geets as being someone who is a very prominent researcher in his field and is kind of already settled in what he wants to do and has already had a negative experience, as was just mentioned, that look, we have this happen, we don't want this to happen again. But it's again the big elephant in the room, because everyone simply knows it happened, but they are unwilling to talk about it in an openly manner. Then we have Dr. Stevenson, who is someone very passionate about what he's doing, given the personal implications that this case represented for him. Let's say he had a family history of a genetic condition called leishmania, where those who have it may die early because they have different tumors around the body, etc. And he had a personal exposure to that, so he is very driven. But at the same time, where should driveness stop? Where will it interfere with your ability to be to conduct research? And then we have Dr. Cameron and Dr. Wong, who are clearly more separate from they think not they separated themselves more from the research. Okay, so moving on, in general, and I think this has been discussed already, what are they trying to accomplish with their research? What is the main goal? And I think it was greatly exemplified when we had the summary, but anyone else wants to participate and tell me what was they wanted to accomplish with this research?
Well, Fleming is raising a hand, and the others, maybe, yeah, anyone?
Anyone? Oh,
Hello.
Okay. Who's talking?
Yeah, I can participate. Adriana.
Adriana, okay, go ahead.
Yes, I guess what they were trying to accomplish in this research, they were trying to get out of drugs, of the HIV drugs, like they want at the end, like cure the patients. I mean, that was the message I get from what they want to trial with this HIV vector. They were trying to genetically change the virus and at the end get kind of cure the patients, like take the patients out of the HIV drugs.
yeah. So and basically what they had was first 9 months phase 1 trial with 5 chronic HIV patients, and we have discussed this. Thank you, Adriana, for mentioning this. And it was basically about viral genetic therapy. And can any one of you, I see some raised hand here, Carlos, can you tell me about the outcomes that they were looking at? We can't hear you.
Please.
Hello.
Carlos?
Can you hear me?
Of course. Please go ahead. Carlos?
Yes. Hello. I think we have to differentiate the outcome here, because I don't know if the study they are looking for a cure, like a sterilization cure, or just a long-term remission. So I want to add that to the comment of the colleague. I have that doubt. I don't know if we are looking for long-term remission here, or is a cure. So I just want to talk about that. Thank you.
Yeah, so thank you for mentioning that. This will definitely become a lot more clear down the road in the course, but let me just talk briefly about it. So basically, as we have different types of phases for clinical trials, we need to have at the same time different outcomes. So for a phase 1 trial, we're mostly figuring out whether it is safe, and if it's safe, figuring out dosages that may work or may not work. So and then in this early on, you don't basically talk about the clinical outcomes per se, but then in a phase 2 trial, for instance, what we're talking about whether the drugs or the intervention works or not, you want to do surrogate outcomes, and then on a phase 3 trial, when we move away from that, you do more clinically oriented outcomes. So that's important to recognize that early on, what we're trying to assess is safety, and whether what's the adequate safety dose that we may choose for a particular drug, if it's a drug or the intervention that we're talking about.
Okay, thank you.
And then there's someone else. Oh, very welcome. Someone else with your hands raised. I can't see your name. I'm sorry about that. It's not showing up in my screen, but there's only one person. So if you know that you have it up, please go ahead.
Oh, I think it's me. I'm Jose.
Yeah, Jose. Yeah.
And about what you were saying, I think their main outcome for the new study is to, as some said in the chat, is to achieve that patients stop taking antiretroviral medications, so they can avoid the side effects of that medications. And we need to differentiate that study goal from the overall goal of Dr. Geek's laboratory, which is to cure HIV. But they are two different concepts in my point of view.
I agree with you. So great. Thank you for your comment, Jose. And then we have different phases of clinical trials, and I'll just talk briefly about them. So we have usually we have phase 1 through phase 4, and we have preclinical studies. And then after the preclinical studies have been carried out, a drug is approved to start human subject testing. And then we move from the phase 1, phase 4. And in phase 1, basically what we're doing to see a particular drug and to see those dosages. We're doing an investigation of a new developmental drug, whether what's the maximum tolerable dose before we get side effects. And then and I wanted to show this because places you will see this, they say we want to of a drug, when in reality, to trial, usually you do not explore the effectiveness but the efficacy to see whether the drug works in a controlled environment. Shital, do you have any particular question on this, or?
Yes.
I see that your hand is raised.
Yeah. So I wanted to know the difference between efficacy and effectiveness with respect to clinical trial.
Sorry, what was that?
I wanted to know the difference between efficacy and effectiveness with respect to clinical trial.
Okay, yeah, we'll talk about we'll talk about it right now. So as I was mentioning, a phase 2 trial, basically we test efficacy, and basically what we say is how the drug works in a controlled environment. Does it really work? It's a proof of concept here. We want to say, does it really work? And then in a phase 3 trial, what we have is we keep exploring the safety, but here what we do is we talk about effectiveness compared to currently available treatments. And effectiveness is mostly, does it work in real life scenarios? Not only in a controlled environment, but does it really work? And here we change a bit the kind of outcomes that we use. In a phase 3 trial, we mostly use clinical outcomes, and as opposed to a phase 2 trial where we use mostly surrogate outcomes and to study. And then after we conduct a phase 3 trial and we get successful results, then this is where we would submit for the FDA for approval. And at this point, the FDA will consider whether the safety and effectiveness of the drug is applicable, whether the results are sound, and if it gets approved, then we move on to the last phase that is known as phase 4, where we are evaluating the drug over time in a large number of patients to assess for post-marketing surveillance. These are large observational studies. These are not where we look for rare side effects or effects that may take time to develop that were not able to be ascertained during the preclinical, the earlier phases of these studies. Anyone has any particular questions at this point? Go ahead.
I have one. What is the difference between?
Yes, go ahead.
What is the difference between efficacy and effectiveness?
Okay. Yes, as I mentioned, whether a particular intervention works in real life scenarios, and effectiveness is whether a particular intervention works in a controlled environment. Let's say, does it really work? That's what we are actually asking ourselves. Efficacy and effectiveness is, does it work in the real world? That's what we are asking with effectiveness. Does that answer your question, Samuel?
Yeah, thank you.
Professor, I have one question.
And then Sebastian.
Yes, sorry. Hello, yes, everyone. I have a question about phase 4 studies. For about how many months or years is usually that a phase 4 trial is developed, or It's like pharmacovigilance when we see a side effect. I don't know, like 20 years ago, the drug was approved. We ran a phase 4 trial that phase. I don't know. I have doubts about it.
So your question is how many years it may take to conduct a phase 4 study?
Yes, a phase 4 trial in order to assume that the drug that was approved is safe. Yes, that's my question.
Okay, so in a phase 4 trial, it's after you have approved the drug, you basically do observational study. You keep monitoring its pharmacovigilance. You want to see what is reported about the drug. So I would say that a phase 4 trial is it's a continuum, and you keep basically monitoring the drug as long as it is in the market for a very long period of time.
Okay, thank you.
Anyone else? I saw.
Yes, I have a
Anyone
question.
else? Someone had another question.
Yes, I have a
If not,
question. Could you hear me? Hello?
Yes, I can hear you.
Okay. My question is about the approval for the FDA.
Yes.
What is the difference between the full approval and
Yes, I can hear you.
emergency use approval?
Sorry, what was that?
What's the difference between emergency use approval and the full approval from FDA? EUA and the full approval.
So you mean the accelerated global?
No.
No.
Emergency use approval by FDA compared to permanent or full approval by FDA.
Yeah, go ahead, Keiko. I'm sorry.
Sorry, I cannot hear. That sound is not very good.
I think,
if you right,
I think I heard. I think I heard.
Please type your word in the chat.
What Samuel is trying to ask is, during when we have developed our vaccines, FDA has issued a emergency approval, and so Samuel is asking, what is the difference between the emergency approval of a specific drug or a vaccine, and how does that approval differ from a permanent approval? As in, I approved a paracetamol for pain relief. So how do they differ? Am I right, Samuel?
Yes, you're right. The difference between the yes. Thank you, Freddy.
So the difference is that the FDA, given the fact that it is needed, there is I forgot the name for it, but there is some sort of statute here in the United States that allows them to bypass certain criteria and allow for the use under the emergency condition. And that was passed as an act. We can definitely share this with you, but and then they don't go through the same process because it is needed, given that the benefits might outweigh the risks associated with not conducting the full process for approval of the drug. In a full approval, well, you go, as it can see here, approximately 5 to 15 years for approval, etc., of a particular intervention. But there's definitely a law that allows the FDA to do that. In certain conditions, it may qualify, such as COVID, for such distinct distinction, basically.
Hi, John. Actually,
So we are running a bit. Yeah.
Sorry, Keiko Tozgin. So just
go ahead, Keiko. I'm sorry.
Yeah, just I found one more question from chat. Rodrigo said, could you please explain a little bit about phase 0? Can you hear me? Can you hear me?
Hello?
Hello?
Can you hear me?
Yeah, now it's okay. The question is about phase 0. Maybe the prior phase of phase 1, I think the preclinical and the microdose or anything like that. So could you please comment on that?
So you're saying, I'm sorry, I think my internet got unstable. You're saying a phase 1 and a phase 2?
No, one student asking about the phase 0. Yeah, some
Okay.
mentioned about the phase 0, but yeah.
Okay, so so the question is a phase 0 study, what that is?
Yeah, what is this?
Okay, so so phase 0 study, usually we can do what is known as a prior to starting our trial, to better get a sense of what we would need. We do some studies to ascertain a pilot study. Sorry about that. I forgot the word. I had forgotten the word. So we do pilot studies, phase 0 studies, to ascertain or get a better picture of what it is that we're going to study and get better estimates of what we're going to study. So we pilot the study, and having some sort of clinical idea, but we don't have real estimates of what we want to do afterwards. So for that, we basically take our clinical ideas and estimation, and then we pilot it in a smaller than usual sample and get a real estimate or an approximation to better understand what the sample size calculation might look like for the next step of what the clinical trial might be.
Okay.
Does that answer the question?
Yeah, I think it's okay. Maybe yeah, one student. Yeah, he cannot talk on mic, so but I think okay. Rodrigo, is it okay? Okay, great. Please go ahead.
Okay, perfect. And then we're moving forward to the ethical part of things. And this was touched upon earlier. And so briefly talking about this, ethics did not actually start until in the 20th century. And here we have seen that, for example, the Hippocratic Oath, that includes the principle of do no harm to the patient, but it was actually not after the World War II that bioethics per se started as a science, and as a consequence of the experiments that some doctors perform on Jewish prisoners in different concentration camps. And after that, we have what is known as the Nuremberg Trial of the 23 Nazi doctors. And then here the Nuremberg Code was developed, and which highlighted the need for informed consent, prior animal work, qualified scientists, risk justification, etc. So this hasn't been such of a linear process, but we have stumbled along the way, because as you see, one negative thing happened, and then some sort of regulatory agency came in, but then another negative thing happened, and again, this has been a continued process. And so I think we talked previously about this, about the basic principles in research, such as autonomy, non-maleficence, beneficence, and justice, which are basically the primary the pillars of ethical conduct of research. And can anyone give me an example of what autonomy means in research?
Can anyone? Oh, I found Sebastian is raising his hand.
Oh, great.
Can you talk?
Yes. Well, autonomy is the faculty of the patients, of the volunteers of the study, to participate in this study and also to drop out from the study whenever they want. So it's like the decision the volunteer have in a research, participate, to drop out, and also to decide whether to withdraw or if they want to complete the research entirely.
Perfect, thank you. And then someone else there, can you talk a bit more about non-maleficence? What do we mean by that concept?
Well, Samuel, you were raising your hand. Can you talk? Samuel? No?
Yeah, Samuel? No?
Yes. Could you hear me?
Okay.
Yes.
It's better. I think it's just do no harm to your patients or to the participants.
Perfect. And then the next concept, let me see, non-maleficence, beneficence. What do we mean by that? Yes, I see. I can't see your name, unfortunately, but whomever has their hand raised, can please go ahead.
Yes, it's me. I have one.
Okay. Yeah, Danielle, go ahead.
Yes. The beneficence is based on the conception of minimizing risk and maximizing benefits for the research participants. So the do no harm comes from beneficence, I think.
Great. And lastly, I mean, we should follow all of these different concepts, but what's the concept of justice in research? Anyone that has not participated, please, do you want to chime in on this?
Many people are raising hand now. So Daimo, sorry, my pronunciation is bad. Daimo, can you talk?
Yeah. Yes. Okay. Justice is about equal and fair selection of test subjects and equal application of testing new drugs or interventions, regardless of any characteristics between the subjects.
Perfect. So we've talked about the basic 4 pillars of conducting research: non-maleficence, autonomy, and beneficence, and justice. Of course, you need to follow all of these: integrity, respect, confidentiality of patient information. But basically, these are the 4 pillars. And so we have different historical landmarks that I want to mention here. This is by no means an exhaustive list, but we know about the Tuskegee study. Basically, the Tuskegee study, it was here patients were treated did not undergo treatment for syphilis. It was a very vulnerable population, although there was a treatment at the moment. Then we have the thalidomide cases wherein patients were given a particular drug, and the deleterious effect that it had on their babies of those patients that were taking the drug were very negative. And then the Nuremberg Trials, as I mentioned at the beginning, basically focused on the post-World War II sequela that happened given the unethical things that were carried out. So we have the Gelsinger case, and I'll go a bit faster here because we're a bit behind. But the main idea was that we had an 18-year-old male with ornithine transcarbamylase deficiency, and we know that this is a condition of the liver where it cannot metabolize ammonia in the urea cycle, and it's usually managed by diet and medication. So this subject was enrolled to participate in a phase 1 study that was basically looking at replacing this particular deficiency that the patient had with an adenovirus that was supposed to serve as the carrier of the modified gene. And then the patient died 4 days later with multiple organ failure from the immune response that his body elicited given the introduction of the virus. And so here there were many red flags, and the FDA determined that the study did not follow the guidelines of informing the exclusion criteria, the potential side effects, and related study resulting in death of monkeys. So they, as we can see here, they were unethical because they did not give the patients and their family or the subject enough information for them to actually make an informed decision about whether it should have been the best for Jesse to participate in this study. And so now that we have mentioned this, I want to ask you a question. What do you think is the impacts of the Gelsinger case in Professor Geek's study? And it's up to anyone who wants to answer.
Hello.
Yes.
It's Fleming, sir. Yeah.
Yeah, go ahead, Fleming.
Yeah, I think first of all, since the Gelsinger case took place in the same university where Dr. Geeks is right now, carries a huge impact that they have to watch out for, right? And they should not tarnish the image of the university twice now. And the second thing is that, again, in the Gelsinger case, it was the novel therapy was related to gene transfer. And again, in this case, the therapy is again gene transfer. So that is why they need to be careful. Again, in that case, they were using adenovirus as a lentivirus. And in this case, they are going to use a weakened HIV virus as a carrier. So yeah, those are the points that they need to look out for.
Great, excellent. And yes, it does have an impact, and is that we are basically learning from the mistake that had already happened. So as we can see, it is a very common topic of discussion during the case that so it happened before. So we should be cautious that not to get very excited about it and to follow the ethical principles. And also, as we can see, it did have an impact on how the university behaved in relation to this kind of studies. So, and here comes a very important concept that has been touched upon in the previous part of this lecture, and is the therapeutic misconception one. And it's basically the mindset on the part of the subjects that they participate in clinical research that assumes the decisions about their care are being made solely with their benefit in mind. And in reality, this is not what happens. And someone greatly exemplified that we do research to generate knowledge, not to treat a particular patient. And this is what happens with the concept of therapeutic misconception. And then I want 3 different people to answer this question. It's basically the poll. If you were the principal investigator in this case, what will your decision be and why? And I see Pedro. Can you go ahead? Hello, Pedro, can you hear me?
Yeah.
Can you go ahead?
Yeah, I was thinking about how the study as it is too risky for subjects.
Okay. Do you have any other particular insight on this matter?
Well, I was just thinking, because the cases of HIV are special cases in this type of investigation. Would it be important to assess all the cases like individually, or how do you select the patients? I think that's a thing that Professor Geeks has to work with his fellows.
Okay, great. And let me just ask you another question here. Do you think in this kind of phase 1 study, generally speaking, do you use healthy subjects, or do you use diseased subjects? And let's just start by discussing that.
Yeah, by the type of patient of that if they have HIV, I don't think that they can use healthy patients. That's why they put it in hyphens, because they are now searching patients that don't require so many treatment, or that they can live treatment depending on the well, to see if they can make a good research. But I think they need to do a better selection of the cases here.
Okay, yeah, but I'm talking like generally speaking. Do you think that when you're choosing selecting the subjects for a phase 1 study, do you choose healthy subjects, or do you choose people with the particular disease, and why?
Well, I think we choose healthy subjects because we are trying to see pharmacodynamics and pharmacokinetics.
Mm-hmm.
So that's the importance to use healthy subjects.
Okay, and in this case, why do you think that they use people with HIV?
Well, the investigators say that they want to have like no medical treatment, and they are trying to find a solution to leave medical treatment. So
okay,
I think that,
great.
Yeah.
Okay, thank you. And then I see someone else, Alessandra. Can you please open up?
Yes, good morning. I have a few questions about it because actually I voted continuous student planning based on the benefits for the society, but as the week went along and all the colleagues with their opinions and the articles that we studied, I have a few questions. First, the subjects are not healthy because of the toxicity of the drug, right? But is that sample size adequate for this particular study? I mean, is there another study that already shown the power of this sample size? Because we are talking about there are few subjects, but there are some studies that shown power enough to show results. So what I mean is, at this point, I don't know the answer yet. I have more doubts than answers. Thank you.
Okay, great intervention. And yes, so there's no right or wrong answer here. You know, and this is the great thing about research. You have to deal with whatever you have at hand and just make a decision based on that. So and yeah, there is the thing about power. Usually, as we discussed earlier, we have small sample sizes when we're talking about phase 1 studies. Given that we're only ascertaining the safety and escalating, if we're talking about a drug, escalating the different dosages to see what's the maximum tolerated or the maximum dose that could be considered considered for that is safe for the subjects. And then one other point that I wanted to touch upon is yes, as you mentioned, Alessandra, we use HIV patients in this particular case, or I'm sorry, the authors, because of the toxicity of the drugs. And we also have other similar scenarios, such as cancer patients, that drugs are very toxic. Might not be as ethical to include healthy subjects in a phase 1 study. So let me just, if anyone has any particular questions at this point, we're going to move to the practical cases now. Please open up your microphone if you have any questions. If not, we'll move to the next stage. No questions? Okay. So first, I want to introduce this great tool, and let me just stop sharing for a minute so that we can go onto that website. It's called, okay, just share again. So this is a great tool with where all the clinical trials that are currently being studied are going to be registered, and we can say for a particular condition, let's say COVID, we can look for a particular condition, and we can see all of the trials that may come up here that are currently being studied, the different therapies. If you have any questions regarding the vaccination, you can find these here as well, for all the, basically, they have to be prospectively registered. And if we go into the advanced search, we can look for a condition, we can select the type of study that is being, we currently want to look for, let's say an observational study on a particular thing, an interventional study. and whether the results have been published, whether they have not been published, and so on. And we can have different eligibility criteria. Let's say that we want to have a particular age group, a particular sex group, and a location where it was conducted. So I would say clinicaltrials.gov is a great resource that could be used to see what is out there, what is currently being studied and has not been published, because as we know, we have the other great tool called as PubMed, but is there what we have is what is published. And many of the studies that have been registered for in clinicaltrials.gov have not yet been published, and many of them also they published a protocol in clinicaltrials.gov, but the results were negative and no paper was produced. So you can get a background or so about on what has been of the what is out there, but it's not published through clinicaltrials.gov. And we'll visit this in a couple of minutes, but let me just first go back to the slides and for us to have some practical
I'm
scenarios.
sorry, can I ask you something?
Sure, go ahead, Alessia.
I have a doubt. So the registration in clinicaltrials.gov is like is like a pre-registration or not? So
what was that? I'm sorry, what was that? I hear some echo and I couldn't get your question.
Yeah, the question is if the registration in clinicaltrials.gov is the same as pre-registration. When you pre-register, for example, a trial or an experiment in a platform, and then you have to follow the phases and the things that you say in the pre-registration form.
So I think that you register for clinicaltrials.gov whenever you're going to conduct a clinical trial per se. This is an obligation for clinical trials. I'm not sure whether I don't think it is necessary for observational studies. It's not mandatory, at least from what I know in general, but it's recommended for you to do it. But for clinical trials, it is a mandatory thing to do.
Okay, thank you.
And then let's get into this. So I'll give you some time for you to read this, and I want 3 volunteers to tell me what phase of study do you guys think it is.
Retrospective.
Phase 1?
Sorry?
Observation.
Retrospective.
It is second phase.
Okay, let me see. Okay, we have let me see. Elmo, can you go ahead?
Hello, it's Elmo from Rio de Janeiro. I think it's a phase 3 trial. It's a randomized trial. Although it's done in volunteers and healthy volunteers, it's a vaccine trial. So we could think it's a phase 1 trial because it's on healthy adults, but it's a multi-center randomized clinical trial thinking about the efficacy of vaccines. So I think it's a phase 3 trial.
Okay, and again, so do you remember what we talked about efficacy and effectiveness?
Sorry,
Elmo?
effective. Yes, of course. So as we're studying, we already have a proof of concept study, a phase 2 trial. So in this phase 3 trial, we are thinking about effectiveness, of course.
Okay, someone else has thinks any different? Anyone else wants to chime in? Fleming, I see your hand is up. Do you want?
After the points that Elmo introduced, now even I'm confused. Initially, I wanted to go with phase 1 trial, actually. Since we are talking about safety and immunogenicity, actually, I'm confused. I would not want to contribute anything. Probably phase 2 trial as well.
Okay. Anyone else? I see someone else with their camera on.
Yes, I think it's a phase 2 trial where they are looking for the efficacy of the vaccine in terms of surrogate markers, which is the titers immunogenicity, or like the titers of the antibodies.
What's your name? I'm sorry, I can't see you on my screen.
Hey, I'm Amit. Sorry, I'm
Okay.
still at work, so.
No worries, no worries. Great. So yeah, you guys have raised great points here. But I think Amit, I'm sorry, hit the jackpot. And as you can see, we don't have any clinically relevant outcome here. And what we have is a surrogate outcome. And we're trying to elucidate immunogenicity of a vaccine candidate, in this case, for the mRNA 1273. And this was a vaccine trial for the Moderna vaccine, and this is a phase 2 trial. And remember, as we spoke earlier, we usually, when we have a phase 1 trial, we're looking for safety, and we're looking for escalating doses of the same of a particular. If we're talking about a drug or a vaccine, whatever, we're looking for escalating doses to see what is the safety cut point for that. And then for a phase 2, we want to ascertain efficacy, and efficacy usually is ascertained through non-clinically relevant outcomes, most likely surrogate outcomes, and still we want to ascertain safety. And then on a phase 3 trial, we want to see whether it works in clinically relevant outcomes. Does it work in the real life situation? So and then we have this one. Anyone else? What do you guys think is the phase of this trial?
Hello, Professor.
Hello?
Professor, I was a little confused that yesterday Professor said in the phase 2 trial, maximum the sample size will be spread from the 20 to 300. But here it is mentioned the 600 in 300 in each arm. So phase 2 in this number of sample size, it is not fit with the phase 2.
Yeah, I understand that. Yeah, and you see, this is where it gets blurry. Although it is the usual thing to happen, it does not mean that it has to be what happens. So I totally understand. You've raised a wonderful point, and it is usually what should happen, but it's not necessarily what always happens, you know? And in this scenario, it is a phase 2. Let me just go back 1 second. It is a phase 2 because, as we said, they're trying to measure immunogenicity of the vaccine, and basically they're asking the pivotal question: does it work? Does it generate immunogenicity? And when we do that, when we ask the primordial question, does it work? That's what efficacy concerns itself with. Does that answer your question?
Yes, Professor.
Great. So this one, anyone wants to chime in? I can't see your name, but your camera is on. I don't know. I'm sorry about that. Well, Rodrigo, do you want to take a
Who? Rodrigo?
Rodrigo, yes.
Oh, no, he cannot talk now. Maybe because he sent me the message. I think his connection is not good.
I can go.
Okay.
I can go if you want.
Oh, Amit. Okay.
Yeah. So this is a phase 3 trial where we're actually, looking for clinical effect in terms of whether the vaccine prevents, active infection from COVID, and it compares between a placebo, people who got placebo and people who got vaccine. So it's we're looking at the clinical efficacy. So this is a phase 3 randomized controlled trial.
Great. And then I see Daniela, you have your hands raised. Do you think similarly? Do you have anything differently?
Hello, I agree. I agree with the last comment. It looks like a phase 3 trial, patients being randomized either to therapy or placebo. It's a big number of patients that are, or subjects that enter the study, and they are testing for immunity or prevention of the infection. So I think it's phase 3. I agree.
Okay, great. And so yes, we can see here where we shifted from looking whether looking at a surrogate outcome to looking at a more clinically relevant outcome and in real life. So it was prevention of COVID-19 illness, and generally speaking is more of a clinically relevant outcome than the previous one. And this is in fact a phase 3 study. And then we have this one, and we have a different question asked with this. What are keywords that may hint at the trial phase with this study? Elmo?
To redeem myself from the previous question, the outcome is more important than the number of enrolled patients. So in the previous study, I just saw the number, and I saw 600 people, and the fact that it was randomized. So the fact that it's randomized tells us that it's not, and of course it's the study is not looking for safety outcomes. Tells us it's not a phase 1, and the fact that we are looking for clinical relevant outcomes, and of course a second hint is just how it's a different one. It's an open label trial, 45 healthy adults. This one is a phase 1. Let me see.
And what else? Do you have anything particular else that caught your attention?
Participants. The fact that it's healthy adults. It's so I was answering about the previous one.
No worries.
So healthy adults, it might be phase 1 or phase 2, because we're talking about clinical trial. Let me see, vaccination. I think it's a phase 2 trial.
Okay. Ava, do you want to chime in on this one? Do you have anything different? We can't hear you. You're muted, I think.
Is it okay now?
Yep.
Yes. Okay. Right. I think the keywords here, it would be dose escalation. It's a small population with only 45 subjects, and they are healthy subjects. And then we end up evaluating the solicited adverse effects. So for these keywords, I would say it would be a phase 1.
Perfect. And that's the correct answer. We see dose escalation. we, as we previously discussed, we're usually looking at what's the maximum dose that's tolerable and its safety for the participants. We are looking for safety and also the small amount of participants. And then the last training case is this one. What phase of this trial? What is the phase of this trial?
Two?
Who was that?
Graciela.
Okay, so why do you think that's a phase 2 trial?
Because they searching to prevent the risk of several adverse side effects.
Okay, where are you seeing that? What part of the abstract?
Wait. Oh no, they don't say anything. Can I change my?
Of course, of course.
Well, I think now is phase 3.
Why? Why do you think that?
Because they use the dose escalations in open label trials, and they was responding, including 40 older adults.
Okay, and so so what does if we're talking about different dose escalation, what they would just have discussed about where is that usually studied in what phase of a trial?
Would it be phase 1?
Phase 1
or 2?
Yeah, phase 1. Usually
and
that's
it's
where
a small sample, right?
Yeah, small sample, your dose escalation, and you're
Open label.
basically looking for, yeah, open label as well. Phase 3, usually you have randomization, usually.
Awesome.
And so we're almost
the
at
primary
the end.
outcomes. The primary outcomes are adverse events.
Yeah, exactly. That's very relevant as well.
Dr. John,
so,
can I chime?
Sure. Go ahead.
Okay, so one thing that I noticed was that in the phase 1 trials, we usually stratify the groups, stratify the whole population, as in the volunteers into groups, and we test different dosages in different groups. Is that something unique to phase 1, or is it something that is interspersed in all the phases?
You could do that. You could stratify it, and should you decide to do it, let me see where stratify. My understanding here is that they wanted to ascertain the safety in two different profiles of patients. Those who were 56 to 70 and those who were older than 71. Maybe they have different characteristics physiologically speaking, and so they might expect a different safety profile depending on age. So that's why they wanted to stratify. But it is not a necessity for you to stratify to be able to do a phase 1 trial. This was simply a characteristic of how this was designed.
So if I see this in a trial, should I be biased towards that it may be a phase 1 trial, or it can be it's an open-ended thing?
No, it's an open-ended
unique.
thing. It's no, not unique.
Thank you.
Okay, so no worries. So we're almost at the end of the hour. Let me just show you again, and then we can take some questions. 1 second. Let me just go back again to clinicaltrials.gov, and we can see. So let me just, okay, 1 second. Okay. Okay, so here we are. Can you see my screen? So this is what,
yes.
Let's say this is the Moderna COVID vaccine. This is a phase 1 trial, one that we just explored. And then what we can see here is that we have a description of the protocol where they say what they're going to do, the summary, description, the type of study design, what the arms are going to be. This is a very useful tool. One, because I mentioned before, we don't usually get, some papers don't get published because if they get negative results, they don't get published for one or other reason. It is not ideal. Everything should get published, even if it's negative, but usually more often than not, those with negative results do not get published. And we get all of the information here about what they did, etc. And then if we go to the top and we go to results, even if it's not published at clinicaltrials.gov, we can see whether a research publication came to fruition for this research. So I would say this is a very important tool for you to keep in mind. And yeah, I'm more than happy to take any outstanding questions still remaining now. And I want to kudos to you all for undertaking this novel course in clinical research. So any questions?
Does arms refers to the different groups in a phase trial?
What was that?
Sorry, arms, you were saying that the different arms, like arm 1, 11, 14, those are groups with different approach. Okay.
Yep. So let's say that we discussed, let me just go back here for a second. Let's say that we have 3 different groups. We have a group that received 25 micrograms, another group that received 100 micrograms, and another group that received 250 micrograms of the dosage for the vaccine. So these are the different groups, the different arms in the trial.
Thank you very much.
Does that answer your question? Okay, great.
Yes, it does. Thanks.
Okay, please unmute yourself if you have any questions.
I have a question, Dr. John.
Yep. Go ahead.
In phase 2 trial, should it be always a randomization or it could be open label?
Most of the time, it is randomized, but you cannot say for certain that it needs to be randomized to be considered a phase 2 trial.
It can be open label.
As you will see, you have, later on, you will see, and I think this was touched upon in the chat, you will see trials that start at phase 1 and then transition to phase 2. This is a bit more different, kind of a different topic, but yeah.
Dr. Polanco.
Professor,
Did you hear me?
Go ahead.
Professor.
Sorry, what was that?
I'm Licia. Professor, if phase 1 trial, for example, the drug for the diabetic, and we are taking the healthy volunteers, and if we administer that medication to the healthy volunteer, how can we ensure the safety? Because the features for the diabetic will not appear in the healthy people, but the drugs for the diabetic patient, for example, some hypoglycemic agent, how can we make sure that it is safe in the healthy volunteers and it will be work in the diabetic patients?
So one thing that you need to consider, and I don't know the profile of the drug that you're referring to, but it could apply here the same concept that we discussed earlier that had to do with HIV and cancer. If the drug is has negative side effects, then you would not use healthy volunteers, but you would rather use those with the disease. Does that answer your question?
I don't clear the concept.
So remember that when we discussed this week's case, we said that the reason why they chose HIV patients as opposed to
That I
healthy.
understood. The HIV and the oncology drugs, it will not be tested in any other patients because of the ethical issue. The same principle is applying for the other medications also. For example, if diabetic medication, it will be tested only those patient diabetic without much complication will be considered as a healthy volunteer in the phase 1 trial.
So it depends on the profile of the drug.
Okay.
If you consider the drug to be very toxic, then and would so it would, like, if you would say, let's say a patient, uncontrolled diabetic patients, then and the drug is a hypoglycemic drug that has a great potential for hypoglycemia, then you would much rather, ethically speaking, consider patients that are diabetics as opposed to healthy subjects.
Yeah, it's clear now, Professor. That's always confusing me. Thank you.
Okay, great. You're very welcome. And then I see
John,
3 hands.
John, sorry.
Yeah.
Elmo here, just to add about safety profiles. In phase 2 and phase 3 trials, although the primary outcome is not safety, you also collect data about safety. So if a drug has not caused any problems in the phase 1, you still can look for safety outcomes in phase 2, phase 3, and of course phase 4 trials.
Totally, yes. Let's see, here I have Rodrigo. Do you have a question?
Yeah, I would like to add on the last comment. Yeah, I totally agree, because if you think like this, you have in the beginning phases you have a small number of participants. So, and as you increase the number of participants, and as the exposure increases, there is a chance like you might see newer side effects and adverse events. So basically these things will be more prominent in phase 3 trials, and also phase 4. So I'm seeing some examples right now at my work. So I'm seeing patients who has been completely vaccinated with COVID-19 being admitted in the hospitalization. And if you remember the the trials, none of the, almost like it was 95% effective, and none of the people who got vaccines in the trial were admitted in the in the hospitals, or they died from the from COVID-19. But we are seeing cases right now. So yeah, that's what I wanted to add.
Yeah, thank you. Thank you. You're totally right, and very important to mention this about the sample size and that in the follow-up times, because as the trials are earlier on in their development, they are not they don't last as much as later trials, and when we do phase 4 trials, which are observational studies. Let me see, we have other question. Let me see, Juan Garzon.
Yeah, thank you. I have a question. Like, as some drugs such as chemotherapy or gene therapy, they need to have like ill patients because they are just very toxic to normal patients. How can we differentiate phase 1 and phase 2? Because if you are just having like ill patients, you'll have the opportunity to check for efficacy. So you are doing the same thing in phase 1 and phase 2. So how can I differentiate? And if you have a very good phase 1 trial, can you skip the phase 2?
Usually no, because you don't usually, so you will learn down the road that you power your study for your particular outcome. And so when you have a phase 1 trial, you mostly looking at safety of the trial, and also if it's related to drugs, drug escalation, dosage, so that you can ascertain whether what's the safety profile of that drug. And then you move on to a phase 2 trial where your outcome will be different. You will still try to ascertain safety, but your main outcome will not be safety, but something related to efficacy. Does it work? And for you to answer that question, you need to know the mechanism of how it works. And so let me just give you a hypothetical example. Let's say that you are using a drug to decrease blood glucose levels, and then you do a phase 1 trial to ascertain what the adequate and safe dosage for that drug is. Let's say you have 10 milligrams, 20 milligrams, 30 milligrams. And then you want to see what's the safety profile of those different dosages for the drug. And then you do a phase 2 trial when you want to see, okay, does the drug work? Does it decrease the glucose level in the patient? So your outcome won't be the safety outcome anymore, but the outcome that will tell you how much it decreases the blood glucose level. And so that's what tells you if it works or it doesn't. And then if you want to continue, you move to a phase 3 trial, you want to say, okay, this a more clinically relevant question might be, or outcome, I'm sorry about it, would be, does it decrease the amount of keto or of let's say of diabetes ketoacidotic episode for the patient? That's a more clinically relevant outcome, and it's not trying to ascertain the mechanism by which the drug works or whether it works, but does it actually works in real life? I hope that answered your question. If not, please let me know and we can figure out other examples that might be more relevant.
It was really good. Thank you.
Okay, great. Ashraf?
Yes, Samir.
Your hand is raised. Do you have any questions?
Yeah, I asked previously, like the question was about the randomization in phase 2, and you answered that. So.
Okay, great. And so we have Fleming.
Yeah, thank you, Dr. John, for the excellent lecture. I had a question which is not related to today's lecture, but I happened to pick up a phrase by professor yesterday. He said about surrogate outcomes, which are seen in phase 2 trials, right? So can you please shed some light on what surrogate outcomes are?
Yep. So we've been talking about surrogate outcomes here a bit, and you will definitely go in depth regarding surrogate outcomes, but let me see. Let's say that we're talking about you want to measure cardiac function in a patient, and you say, okay, so how do I measure cardiac function in a lab? No, let me jump. Sorry about this. Let's talk about myocardial infarction. Myocardial infarction you can measure in different ways. You can say, okay, surrogate outcome for myocardial infarction would be a troponin level. That's a lab value. That's your surrogate outcome. And then more clinically relevant for myocardial infarction is mortality of myocardial infarction. So that's a clinical outcome. And then you have a surrogate outcome that is surrogating for that myocardial infarction through troponin. And what we're doing here, and I think with these examples that we've had, let me go to a phase 2, to the first one here. So immunogenicity of the vaccine is your surrogate outcome here. And then your clinical outcome is prevention of COVID illness. Surrogate outcome is something easier and shorter in length for you to measure than a clinically relevant outcome. And it's usually a measure that is supposed to some degree correlate with a clinical outcome. But it's not a clinical outcome. It's simply mostly used to ascertain efficacy.
Thank you.
Does that answer the question?
John. Yeah, that was really good.
Great.
Can I also ask, do you measure both of these as in the surrogate outcome and the clinical outcome in the same study, or do we have separate individual studies to measure the surrogate outcome and the clinical outcome? As in,
so
if we were,
Go ahead. I'm sorry for interrupting.
Okay, so like the first example that you gave regarding troponins and MI, so we would not really reach a position wherein the MI will lead to death, right? And we are only measuring for the levels of troponin in a person who has a myocardial infarction. So in this case,
yeah,
in this case, it won't
in this
go
case,
in hand to hand. In this case, that the surrogate outcome as well as the clinical outcome should not or would not go hand to hand. These will be separate entities.
So I will point you towards Bradford Hills criteria. The Bradford Hills criteria are a set of criteria that say whether an outcome is a good measure or a good, let's say, bridge between what is supposed to be the clinical outcome and the surrogate outcomes. So as I mentioned, usually we use surrogate outcomes when we don't have time for an event to occur. So we'd say, okay, we want to see whether it works, but we don't have the time for the event to occur. And that's why we usually have surrogate outcomes when we do phase 2 trials, meaning when we are trying to see the efficacy, when we are trying to answer the question, does it really work? And then we do a phase 3 trial when we mostly, and I don't want to say always, use a clinical outcome, a more relevant outcome that does it really work in real life scenario. So so to answer your question, in short, I would say no. You do not necessarily need to measure both of them at the same time, but I would point you towards the Bradford Hill criteria.
Thanks a lot, Dr. John. That was perfect.
Okay, great. Let me see who else we have here. Cindy.
Hello, thank you. Is that correct? I thought about the surrogate outcomes. I think keywords that we could use to identify the surrogate outcomes would be, for example, biomarkers and numbers and laboratory outcomes. I think Professor Fragney said something about it yesterday, maybe, and that we should think about them as numbers. How can we put our result into numbers? So I think that way it would be easier for us to identify what would be a surrogate outcome in a specific study. Does that work? Is that right?
Yep, that's perfect. Perfect. Thank you. Rodrigo, do you have any other question? No. So if nobody else has any other question, thank you so much for attending today. I wish you a great success during the course, and thank you for your great participation.
Thank you, John. It was great.
Thank you.
So many questions. Thank you so much, everyone. And everyone, we will have another great lecture about the outcome and the research question, and we can learn. So maybe, but if you have remaining question, maybe we will send the email to us, and we try to answer. I was trying to answer to your question, but it was not possible because I saw so many questions. So thank you so much, everyone, and thank you so much, John.
Thank you, everyone. Thank you, Keiko.
Great lecture.
Arigato.
Thank you.